So I'm going to start recording. We're waiting on somebody. No, that's not what I need. Okay, I'm going to share the screen. So I'm going to go to, I'm in Canvas here, and I'm going to go to the syllabus that I uploaded a little while ago. See if it'll open. Well, maybe not. There we go. So here's spring break, and here's where we are. Okay. I think it's assignment 7 that's posted out there. We'll look at that in a second. So we're going to do some spectroscopy. We're going to start with absorbance, briefly discuss fluorescence, and then do circular dichroism. Okay. The material is these 2 chapters out of an old edition of Van Holde, chapters 8 and 10. The new edition of Van Holde, the one that has the 3 authors that you have a PDF of, it's in the same folder. Well, maybe it's in that, it's in a folder close to this one. The spectroscopy has been expanded pretty dramatically. And a lot of quantum mechanics has been introduced, so it would have taken us a lot longer and been a lot more challenging to go through those chapters. I'll point out some things primarily at the end where they make a lot of improvements, if you will. Okay. The way this is written on the 29th, we're going to discuss a couple of papers on extinction coefficients and light scattering corrections. I think we're going to get through that today if I am efficient today. So I think we're going to get through this today. And then the next topic that's listed out here at the moment is gel electrophoresis, polyacrylamide gels. SDS primarily and isoelectric focusing. And there are some classic papers out there that I like to go over. There's also a chapter from an old Van Holde that we'll talk about in terms of some basic and fundamental stuff. The thing that I want to point out is that a lot of you are sort of late with homework assignments and struggling with some of the homework assignments. So I think whatever we don't finish today will carry over to the next time, but it won't be very much. And so what we're going to really do probably on the 29th, next Tuesday, is go over some of the issues with the homework assignments and try to get everybody up on the same page with the homework assignments. Okay. That number 5 that everybody is struggling with, but many of the other problems as well. I have some things from you, but I don't have everything, obviously. And so we want to see if we can get you up to date. The philosophy of this class, which I probably have stated in a couple of different ways, is that I lecture on some basic concepts, beginning with pH and statistics. But then I get into thermodynamics and then primarily binding and applications of some thermodynamic concepts to binding. And the way I evaluate you and help you learn material is to give you homework assignments, problems, which are primarily math-based. That's going to change a little bit in this final section. I'm going to ask you to write a couple of summaries of papers. And this next homework assignment has a couple of descriptive things in it. Here's some data, what's going on, and how would you interpret these data? Because in the end, that's really what this is about. This is about what's the question, what's the hypothesis, how do you answer it, what techniques allow you to draw conclusions. And so that's really where this is going, and showing you how thermodynamics and binding and a lot of the techniques that we're talking about are useful potentially to characterizing of macromolecular shape, size, interactions. Okay, so I think that's probably the way this is going to go next week. I haven't actually outlined how that's going to happen. We'll look at various problems from the homework and go over some stuff and try to get everybody up on the same page in that regard. Then we'll do electrophoresis, and right now the schedule is talking about these videos, which I made 2 years ago when we went to online in the middle of the semester, and turn these into discussions. I'm probably going to remake these videos in some ways to achieve the same goals and get us up to date. I may have you fit data. The problem is that the software packages that I use are PC based, and I know at least 2 of you are Mac based. And so they won't work on your Macs unless you have some kind of an emulator, a DOS emulation. You need to be able to run Windows, basically. So unless you have that, it makes it difficult. It's possible we could set you up on a computer to be able to do this, and that may be an ultimate solution. Okay. What we're moving into is sedimentation, hydrodynamics. Often I cover diffusion as part of this, and I'll kind of count the days and make a decision about that. But primarily it will involve an introduction to the various ways that we think about and analyze centrifuge data. I think what I did this 2 years ago was put it all into uploading software and then giving you assignments. I need to add a serious lecture about the foundations of sedimentation. And then what I like to do is cover a series of papers on sedimentation velocity, a series of papers on microtubule assembly. These papers also have a lot on thermodynamic interpretation of data. I usually do a series of papers on Vinca alkaloid induced tubulin assembly. And in previous years, we were done early May. And so obviously that's not going to happen this year. So around this time is when we'll step backwards and we'll bring in some thermodynamics papers that have to do with drug discovery. And I'll make that much more clear here. I've done that in the past, and that will sort of be a series of introductory concepts that help us understand this. And then there'll be a couple of weeks for all of you to make a presentation. We'll see if we can do it in 2 weeks. Okay, so that's basically the outline at the moment for where we're going. And I'm pretty sure these 3 series of papers will happen simply because they're really nice introductions to basic concepts, but from an actual published paper. And you can see some of this work involves my work, but this is an extension of the Friggin and Timoshev. This paper is basically a reworking of the same stuff that Lee and Timoshev did in a more quote unquote modern form. This was also a Timoshev paper where we basically worked on the same project for 15 years. And so it'll be a continuation of what Timoshev did. Hopefully a vast improvement, and you'll see that. So that's sort of where we're going. Okay. Okay, I also included chapter 5 as a reference for this lecture, but it's basically context. I don't refer to chapter 5 really very much. Okay, so we're down here in the absorbance. So there's the reference to Creighton. Here are the 2 chapters. We're going to talk about this paper, which has to do with predicting extinction coefficients. We're going to talk about this paper, which has to do with correcting light scattering. There's the PowerPoint. And then here's the assignments. And if at the end of all of this, I'll talk to you, maybe at the end of the lecture today or at the end next week. So let's bury that. Let's bring this up. Okay. So this again summarizes what I just said, and notice it's general background, and this is telling you we're going to use this to predict the protein extinction coefficients, corrections for light scattering. And when we're doing this, I may open up this software package to show you something about how the software that we use is usable or useful from a sequence, basically, basic information about a protein that we need. We primarily need that in the hydrodynamic data in the sedimentation part of the class, but I want to show you that application. That software also only runs in Windows, so it won't run on a typical Mac unless you've uploaded some other software to emulate Windows. Okay, so chapter 8 begins with an introduction to electromagnetic radiation. And this is an introduction to a fundamental physics concept that light can be a wave or it can be thought of as a particle. Okay, and when it's a wave, it is basically being scattered. And the rules of that scattering come up in a light scattering lecture that we probably don't have time to go over today, or in this block, in this course. But basically, the scattering of light reflects a property of light that is wave-like. There's probably supposed to be a hyphen there in wave-like, or a space. An easy summary of this is, why is the sky blue? Can you tell me why the sky is blue? Anyone? Children ask, Mommy, Daddy, why is the sky blue? So we're talking about scattering. So it has to do with scattering phenomena. It has to do with the fact that light is actually a combination of all wavelengths. And those wavelengths enter the atmosphere and are scattered. And the scattering has certain characteristics. And those characteristics involve wavelength dependence. And so certain wavelengths are scattered more or less depending upon their wavelengths. It goes as usually the inverse of something like the fourth power of the wavelength. That will come up in the scattering correction we talk about in absorbance corrections. Okay, and so blue light, it turns out, is scattered more than all the other light because of its particular wavelength. And that's what we see in the sky. The other light is being scattered too, but just not as much. And so we get an enrichment in blue light. And then that's what we see in the sky. Except at sunsets, when you're looking into the sun and suddenly it's red. Why is it red? What happened to the blue? The blue is scattered, so it's no longer now on a path to your eye. The red is scattered less than the blue, and that's what you see, a predominance of red light. Okay? And so that's physics, that's atmospheric science. but it's basically the same concept as what we would talk about in the wave properties of light and how light is scattered and what its wavelength dependence is, okay? Otherwise, light is actually a particle, and this has to do with the fact that the light can be absorbed by whatever it's interacting with, okay? So when light hits a particle, partially it's scattered, but it also might be absorbed, okay? And so this simple dichotomy is a reflection of the physics that electromagnetic radiation is a wave and a particle. And we can think about it as a wave, or we can think about it as a particle. Now, to really understand this and to go back to where this sort of came from in the last 100 years, you basically have to talk about this in terms of quantum mechanics, okay? And so we'll do a little bit of that at the beginning, but the fact is that that's really what you need to think about and talk about in terms of absorption and why light interacts with particles the way that it does, okay? So you need a quantum mechanical understanding of this. And that, as I say, the other edition of Van Holde gives you an introduction to that. When Van Holde got two other authors, Johnson and Ho, they're the ones who brought that sort of to the table and gave an expanded treatment of that. So what we're really interested in, though, is understanding how light interacts with biological molecules and the chromophores that might be associated with a biological molecule, and what we can learn about those molecules because of the interaction of light with molecules, okay? And so that's sort of the overview of everything we're about to do. Okay, so we're going to begin with absorption. Let me find that other chapter here, which I don't really need as I go through here, but we're going to begin with absorption. Okay, and
Yeah.
Yes?
And can you once again say what is chromophores?
Heme, vitamin A.
Okay.
Okay, it's groups that bind to macromolecules in some way, shape, or form. And they're usually called chromophores because they often have color, or they often interact with light in a very particular way because their electronic chemical structure causes them to interact with light and give a characteristic absorption spectra, for example, or a characteristic infrared spectra, for example. Okay, so they're the ligands that bind to macromolecules. Okay, in medical biochemistry, we would think of this in terms of vitamins. We would think of this in terms of factors that help enzymes function. Okay, in this context, we're thinking about things that have particular interactions that give you a unique window into the chemistry, the chemical environment of the macromolecule. Okay. All right. So one easy, inaccurate way of thinking about this is that absorption will occur when the frequency of the electromagnetic waves are close to or equal to the natural vibration frequency of whatever the macromolecule or molecule or chromophore is. All right. This is inaccurate because it doesn't entirely explain everything. Okay. It was the way that we thought about this 100 years ago, 120 years ago, until it was all thrown on its head by quantum mechanics. This does not explain sharp spectral lines nor the amount of energy absorbed at any given frequency. That's what we're going to talk about next. So we must introduce quantum mechanics. So the textbook, and I just copy pasted this from the textbook. Let's get this guy completely out of the way down here, completely out of the way. So in the book, it introduces quantum mechanics in this way. okay, and showing that it differs from classical misunderstanding of what light was. So the first thing is that energy is basically absorbed, but only in certain ways. It has to be absorbed according to this sort of relationship, where the absorption is related to the frequency of the radiation by this Planck's law, where the energy is equal to Planck's constant times the frequency. All right, there was no expression about this in classical physics. So it introduces, sorry, I needed to pull something else out that I didn't have out yet. It introduces this particle aspect on radiation. It makes it obvious that there's some specific energy associated with the light that can be reflected in this relationship. And then the second recognition is that atoms and molecules can exist at only certain energy levels. This turns out to be a natural consequence of light being a wave, okay, or particles being represented as waves, all right? Another way of saying this is that energy is quantized, okay? And so quantum mechanics quantized. And so these two phenomena, or these two statements, express at its fundamental level the particle wave-like properties of molecules. So the simplest introduction to this is what is known as the particle in a box, okay? And so in classical physics, a particle can have any energy. Why shouldn't it have any energy? In quantum mechanics, constraining a particle restricts the energy to certain quantized level. So there is that quantized phenomena. I'm gonna have to push you guys even lower here. Okay, so the idea is that the energy is quantized into levels. The levels correspond to the n factor. Okay, so it's n squared. The h is the Planck's constant we talked about up above. And then it's divided by the mass and a squared, where a is the dimensions of the box, okay? And so the amount of energy will vary as 1 over the molecular weight and 1 or the mass of the whatever the particle is, and 1 over the box size, one-dimensional box size squared, all right? And so you can see that if n is it's n squared. So if n is 1, 2, 3, 4, 5, this is the way that the energy will be quantized. On the previous slide, or a slide above that here, it said this does not explain sharp spectral lines, nor the amount of energy absorbed, okay? So the sharp spectral lines come from this quantization of energy, because you'll get a line that corresponds to 1, 2, 3, 4, 5, the energy at which the particle will exist if it's energized up to those levels, and those will give you the lines that exist, okay? And so a lot of this originated in the fact that when the physicists were taking spectra, they could see these sharp lines and they didn't know where they came from. And this was the introduction to solving that problem. Okay, so the particle in the box gives you an introduction to quantum mechanics and explains where the different energy levels, E sub n, come from. All right, now there's a lot more to this. That's sort of a very crude sort of introduction to what we're talking about. Now, absorption will only occur for frequencies we said up above. The way it was set up above was close to or equal to the natural vibration frequency. Natural vibration. Okay, natural vibration is sort of vague. It doesn't really explain where it comes from. Okay, the natural vibration comes from the fact that transitions can only occur between various quantized levels. All right. The levels depend upon this spacing in the quote unquote box. Okay. Now think of the box as the size of the molecule that we're talking about. All right. So in this particular example, okay, they're talking about this essentially, it's not a dimer per se, it's a molecule that has two centers, they might be oxygen, for example. Okay, so it's a diatomic molecule. All right, and this diatomic molecule has a certain spacing between the atoms, but that spacing also reflects certain sort of natural vibrations between that distance, or natural twisting motions or natural rotation motions. Okay, and all of that ends up showing up in what are called electronic, vibrational, or rotational levels. All right, so it has to do with the fundamental interaction between these molecules. So it has to do with some kind of a binding interaction. The real fundamentals of quantum mechanics are what are those binding orbitals, what do they look like, and how do they interact? And then there might be something vibrational. This might actually be oscillating. And then they might actually be rotating relative to one another. And as they spin and tumble and rotate, that gives rise to another sort of interaction. All right. And so these interaction levels that are depicted in each one of these figures reflect the particle in a box sort of idea. But then you can also have what is thought of as a ground state and an excited state. Okay. And so these two electronic states differ in their potential energy and in the position of the minimum in the equilibrium between these two atoms. And so you have these various levels, and the energy is therefore absorbed when there's a transition from one level in the ground state to another level in the excited state. And so the frequency with which this happens depends upon the energy of this transition. The transitions occur between electronic levels or vibrational rotational levels. Okay, and so the vibrational levels tend to have smaller spacings. The rotational levels also tend to have smaller spacings. And so these transitions reflect these various levels and energy being absorbed to drive the ground state into the excited state. And so these are what dictate basically the absorption characteristics of this molecule. Okay, and the text over here in the legend explains a lot of this in the sort of the way that I said it. Okay, and so notice electronic states, different levels of vibrational energy, the long lines, and the rotational energy, the short lines. All right, and so various kinds of transitions can occur, and that's what's reflected in what the absorption spectra is going to look like. Okay, and so the long line going up is the electronic transition. A vibrational transition occurs in this transition here between the lines. And so the amount of energy is therefore inversely proportional to this spacing. Okay. Now, in terms of thinking about this, you need to relate the wavelength of the transition to the frequency, which gives you some connection to the amount of energy. And so you need to think about the fact that the wavelength is inversely proportional to the energy. All right, and so long wavelengths would be lower energy and short wavelengths would be higher energy. And that's sort of what we're getting at here. And so when you have a plot involving wavelengths going from, notice here, 100,000 nanometers down to 1 nanometer. What we're talking about are X-rays with the most energy, and then the ultraviolet region, which basically goes up into the 300s or so, above the 300s, and then a visible region, which is the light that you can see, blue light, red light that I mentioned. And then you get into something called infrared, and then you get into something called microwave. And relating this to the transitions that are occurring, the most energy is occurring for the electronic transitions. These correspond to inner shell electrons. And then you get up into the visible, and this becomes something involving outer shell electrons. And then you get up into the thousands of nanometers, and this is vibrational transitions. And then tens of thousands, you get into rotational transitions. And so depending upon where you're looking, depending upon what wavelength you're using, you are basically looking at various kinds of transitions that are occurring in these molecules. All right. And so this part of the chapter sort of gives it on a mathematical footing. It basically tells you that the vibration, the frequency is equal to the wavelength of, the speed of light divided by the wavelength. This ends up being in units of inverse second. Often people express things in terms of wave numbers, which is 1 over the wavelength without the speed of light. Ultimately, what we sort of want to do is express this in some way that expresses something about a fundamental property of the molecule. And so we do this with something called Beer's law, which reflects the intensity of the light. It's not just the wavelength, but what its intensity is. It corresponds to some intrinsic intensity going into the solution, for example, or going through the molecules. And it's reflected in some relationship involving an extinction coefficient. This is this epsilon term, a length, which is the path length through which the light goes through the solution, and then the concentration in some units, okay? So the intensity transmitted through a cell of thickness L centimeters containing a solution of certain moles per liter will give you what the output intensity is going to be. So you're going to have some amount of absorption, obeying these rules, corresponding to these particular transitions, and the amount of energy that is absorbed, or the amount of intensity that is absorbed. So the decrease in the initial intensity will correspond to this equation, epsilon LC. So higher concentration, longer path length, and an intrinsic property known as an extinction coefficient. If you change the units here and bring I0 to the other side, then this becomes percent transmitted light. Okay, I over I0, how much light comes through? If the concentration is too high, nothing comes through. You can't see it. If the thickness is too long, you also can't see it because a lot of the particles get absorbed. We can then define this as the log of notice we did the inverse here, the log of I0 over I. This is defined as the absorbance. And when you take the logarithm of these equations up here, what you end up with is that absorbance is equal to the extinction times the path length times the concentration. Now, the units of the extinction depend upon the units of the concentration. So if C is expressed in grams per 100 mils, that's essentially a weight percent concentration. And so the extinction is in weight percent. Okay, if we express C in terms of mgs per mil or in terms of moles per liter, then the extinction coefficient will have the appropriate units. All right. And so this is the fundamental thing that you sort of need to glean at the beginning. It boils down to this equation. All of this is sort of prelude to absorbance is extinction times length times concentration, path length through the solution. This is obeyed in all of these different scenarios, but at different wavelengths, you will have different extinctions because there will be different kinds of properties that are being activated. On the right is an example of a copper protein called hemocyanin. Okay, and it's showing you what the extinction is. So this is basically, assume you have a 1% solution, 1 gram per 100 mils. And this is the various wavelengths. So it's showing what the extinction would be for various wavelengths for this copper protein. Okay, and so these bands at near 600 and in the 300s, these bands correspond to the copper. The aromatic side chains correspond to this peak here, which is basically tyrosine tryptophan. And then everything down below that in the low 200s corresponds to the peptide backbone. Okay, we'll talk about all of this again later. And so taking the absorbance of this protein potentially gives you information about the copper, tryptophan, tyrosine aromatics, and the peptide itself, okay? Now, what we normally do with this stuff is measure the absorbance, and knowing the extinction coefficient and the path length, we can determine a concentration. So we use this kind of information usually to get concentration because we know what the extinction is, all right? Changes in the shape of these spectra potentially give you information about changes in the environment, which may reflect changes in different kind of vibrational states or different kind of electronic states or rotational states. Information about maybe the changes in the binding properties. Okay. So,
Dr. Correa,
yes.
I'm sorry, I just have a quick question for the spectra on the previous slide. I'm just curious, is that like a known thing that if you see, I guess, how do you know that the absorption that you see for copper is copper and maybe not another?
Well, that's part of the whole thing. I mean, this is a known in this particular case. We know that hemocyanin has copper bound to it. So that partially comes from molecular characterization of hemocyanin. So obviously it has to be sort of broken down. We probably use some kind of mass spec to get the identity of the copper. Okay, the fact that it's a protein, we probably know the sequence. So we know something about the aromatics and the backbone, but the presence of the copper is because by some other technique, we have identified the copper. Okay, it has a certain color, so you can see that partially. This is the visible part of the spectra. So copper will have certain kinds of colors depending upon what it's bound to. That will distinguish it, for example, from iron or other kinds of metals. And so that all has to come from other kinds of information. Okay, when somebody first isolated hemocyanin or first isolated a solution of homogenized, whatever they homogenized to get this, they could see these bands and they could say, gee, I wonder if there's a copper containing protein in here because we've already done fundamental experiments on copper containing molecules. Okay, does that partially answer your question?
Yes, sir, that makes sense. Thank you.
Okay. So a typical spectrophotometer, okay, and so that's what this is. It's a outline of a simple spectrophotometer. Most of you have a spectrophotometer in your lab. I know a lot of you use other kinds of devices, or you don't actually use spectroscopy for determining a lot of concentrations. But a typical spectrophotometer, I've got 2 of them in my lab at the moment. I used to have 4, but I just got rid of 2 old ones that I had. Has some kind of a light source, okay? And so you have to have some kind of a light source. It's usually both a visible and a UV lamp. And then you have to have some way of selecting wavelength. So there's some kind of a monochromator that allows you to select a particular wavelength or a narrow band of wavelengths. And then that light is shot down a pathway. It's usually split into a sample and a reference. So this would be a double beam instrument. double beam to subtract the reference due to solvent. So I show that up above, okay? And so we generally have two cuvettes, one for the sample, one for the buffer. Both of these impinge upon a photomultiplier. We take the difference and we plot out what the absorption spectra looks like. So what generally happens is that there's a wavelength, there's a white light, goes through a monochromator, and then we scan wavelength, usually from high to low. Okay, so you might go from 600 to 200, and you do that at every single position. Notice by the light being split, the initial intensity going in here is the same in both samples. And so taking the difference to generate the log of I0 over I makes sense. In a single beam instrument, you put the reference in, you take the spectra, and then you put the sample in, you take the spectra, but then you subtract it by computer. And so if the I0 is changing, then it's not exactly a perfect match, if you will, okay? Generally speaking, that's not a big deal for just concentrations, but for other kinds of things, it can be a big deal. The variation in I0 can vary enough to cause trouble. So this is a basic outline of how it works, okay? Variation in wavelength by some technique, sample minus reference gets you an absorption spectra. Okay. I'm not going into all of the other aspects of this, the selection of wavelength in the monochromator. Most of the good ones use a grading method. You can also use a prism to select wavelength. Conceptually, you've seen pictures of prisms splitting wavelengths, splitting light out into various colors. And so that's basically what the prism is doing. And you're kind of tilting the prism to get the different wavelengths. The grading is a more accurate way of usually doing it, but we won't go into all of those details. Okay. So the next section is on infrared spectroscopy. At short wavelength regions, we're looking at vibrational transitions. So look back at that previous figure. So a nonlinear molecule will have n atoms. There will be 3n minus 6 fundamental modes of vibration. This has basically to do with sort of thinking about how multiple atoms might interact with one another and how they might vibrate relative to one another in a structure without trying to map it all out and figure it out. Different ways of combining vibrational frequencies. Okay, and so there'll be a whole series of modes that you can in principle activate so that you can absorb that light and study specifically that vibration. All right, and then they give examples of this, and this is where it begins to kind of make maybe more sense, that if you have a carboxyl, okay, a carbonyl, excuse me, if you have a carbonyl and a fundamental stretching frequency, you discover that this occurs at 1700 inverse centimeters. All right, and so notice this is plotting wavelength and it's also plotting wave number. Okay, and so here are the positions of those frequencies, and you can see that that's where you can expect transitions. And so you can be looking in this region for changes in the environment of this carbonyl. All right, and then you can see that things like C double bond end or phosphorus groups are in adjacent positions. But you can see that all of the typical groups that we might see in a macromolecule, a hydroxyl, an amine, a CH, an SH, all of these are accessible at different wavelengths, at different wave numbers. Oh, and notice they're replotting these for hydrogen bonding and non-hydrogen bonding. So there are transitions that occur. And so this is the value of infrared, that it tells you something about the environment. It tells you if it's involved in a hydrogen bond, and you'll have shifts in the hydrogen bond depending upon the interactions that it's undergoing in the molecule. I think I say this down below, but notice these big white stripes. This is water. And so water is a big problem in infrared because water actually absorbs in these regions. And so that you can see that it interacts with hydroxyls and carbonyls, and it makes it difficult to make the measurements. If you've ever been in a lab that does infrared, often what they do is they make a powder and then they press the powder into a sheet, usually in some kind of a cell that then can be mounted in the spectrophotometer. And so what you've done is you've gotten rid of the water and you're taking an infrared spectra on a solid solution of whatever this molecule is. All right. And so it avoids the water problem. Okay, so this shows an example of poly-gamma benzyl L-glutamate in alpha helical form. And notice it's a solid film of the polymer, just as I just described. It says it's been prepared and oriented by stretching. So what they're trying to do is get all of the helices to line up. This molecule, poly-gamma benzyl L-glutamate, is known to make oriented alpha helical groups. And depending upon how you prepare it, the entire layer will be all pointed in the same direction, but the layer above it will be slightly oriented and rotate. And so you can study a lot of really interesting things, but if the layer is thin enough, then you can shine polarized light along the axis or perpendicular to the axis. And when you do that, you can see different absorption characteristics, reflecting the fact that the shape of the orbitals that it's interacting with, the shapes of the bonds that it's interacting with, make the light more or less absorption capable, depending upon the light is interacting down the axis or perpendicular to the axis. We're going to come back to all of that by the end of the lecture and in the CD lecture. Okay, and so you can see that the carbonyls are decreased in their stretching frequency. We said up above that it's 1700, but here obviously they're involved in some kind of interaction. And so it drops to 1652. I think up here they drop slightly. Okay, so it's only a small change, but it's a noticeable change in the spectra because we can do the wavelength very accurately. Okay, and so all of the fundamental transmissions, how much light is being absorbed, and therefore less light is being transmitted. That's why the percentage goes down. The I zeros are up here near 100%. And so it goes down. So the light is being absorbed at this frequency corresponding to this group. Okay, so you are specifically probing the environment of the carbonyl, specifically probing what it might be doing. And then you're also looking, because you can make it a solid film and do parallel perpendicular, you're also learning something about the orientation of the helices relative to one another. All right. So this is the kind of information that you can glean from something like infrared spectroscopy. Most infrareds, I'm not aware of one being here on campus. There's a device over in Belhaven in Reed Bishop's lab, and he will make films on a lot of stuff. He will compress the sample into a dried film and then take the spectra through the film. Here's an example of the hydrogen bonding signal. Remember up above, it was showing you the difference between non-hydrogen bonded and hydrogen bonded transitions. Okay, so it affects the electronic configuration. This is a sample of a study where they were mixing, let me go ahead one slide. They were mixing cyclohexyl uracil and 9-ethyl adenine in a deuterated chloroform solution. Deuterated because we don't want to look at the hydrogens in the chloroform. We want to look at the hydrogens in the molecules that we're interested in. Okay, and so they made mixtures of these solutions. 100% U, 100% A, and then combinations down to 50-50. And they looked at particular changes in the IR spectra. Again, transmittance, so the light is being absorbed. Okay, and then they plotted the peak position at various wavelengths, okay, as a function of the mixture. And what they found was that at 50-50, there was a peak corresponding to 3330 or 3490 or 3260. Okay, and so this suggested that there was a 1-to-1 mole fraction mixture, that there was a bonding occurring in a 1-to-1 ratio corresponding to these different wavelengths, which give you information potentially about the groups that are involved in the hydrogen bonding. Okay, and so this was basically a really early study. This is 1965. This comes from the lab of Alex Rich, a real early study on the fact that uracil and adenine hydrogen bond to one another. Now that's something you just know. You have been taught that now, right? That U and A hydrogen bond. Well, somebody had to discover that. Right? Somebody had to establish that the U and A is a 1-to-1 hydrogen bonding. Now, by 65, we certainly already knew that from other studies that were done by other people, including Watson and Crick's application of that. But basically, this is sort of fundamental evidence of that. All right. Okay, and so this is another example of what you could use IR for. Now, I want to take a moment to do something, because I've said this to you before. You guys know stuff from textbooks, but I keep wanting to make it clear to you that everything in textbooks came from papers, and everything in papers came from the work of individuals who focused on stuff for some reason. All right. So Alex Rich, this is sort of taken from his wiki page, worked with somebody named John Etzel. And most of you won't know that name, but John was a really influential biophysicist, created a great department at Harvard in the 40s and 50s. And Alex worked with Etzel when he was an undergraduate at Harvard. He then did a postdoc at Caltech with somebody named Linus Pauling. Presumably, you all know the name Linus Pauling. He's famous for all kinds of stuff in terms of especially some weird ideas about vitamin C. He there met Jim Watson, okay? And then he actually went to the Cavendish in Cambridge when Watson was working with Crick, and he worked with Crick while he was there. So partially this is a who's who of, who are these people and where do they come from and where do they get their influences and where did they learn what they learned and how did they apply it? He spent his entire career at MIT in Boston. So he's known for discovering stuff about nucleic acid hybridization. Specifically, he's known for the fact that RNA can hybridize with DNA. Okay. With Crick, he solved the structure of collagen. He's also the guy who discovered polysomes. So you all basically know what polysomes are, and you know what ribosomes are, and you know that they read along messenger RNA. Well, he in early 60s discovered polysomes. He is also the guy who discovered tRNA structures. He crystallized, excuse me, a lot of the tRNA structures. And he's also the guy who discovered something called zDNA, which for many decades was a pretty important and interesting molecule. I don't know if we talk about zDNA anymore as much as we used to talk about it in the past. Okay, we used to think that there was some magical function for zDNA. I'm not sure if we still believe all of that, but it was a different form than bDNA. Okay. Now this next, so I did that, I'm sorry, I did that to make you aware of the fact that a lot of the names and the people that we're talking about here have a remarkably rich histories and contributed a lot to our fundamental understanding that in textbooks basically just gets presented as facts. Okay, and so you need to be aware of the fact that somebody discovered this sometime. All right, now figure 8.8 is introduction to something that I set up above, and it has something to do with the angle at which light interacts with the molecules. All right, and I said you could take parallel or perpendicular sort of spectra when you use polarized light. So that would be light that is a wave, but it literally only has a wave in one dimension. It's not all dimensions white light or incoherent light shining down on the molecule. And that polarized light has this one electric vector that can therefore interact with a molecule that, if itself is oriented, will have an angle of interaction. And that angle of interaction will show up as the absorption relative to perpendicular or parallel. Okay, and it will go as the cosine squared of the angle. Okay, and so that's important because at certain angles there will be no absorption. At other angles there will be maximum absorption. And that's basically what happened in this type of a spectrum, okay, where the angle between the oriented alpha helices and the electric vector of the polarized light was such that there was more absorption in one dimension than in another dimension, okay? And so notice it determines the probability of absorption. The introduction of the word probability there is really a reflection of quantum mechanics and the fact that in quantum mechanics, everything ends up being a probability of something happening, not an absolute, as it was in classical physics. Okay, so the transition dipole moment makes an angle with respect to the direction of polarization of the vector. This is for an oriented sample. The energy absorbed is therefore proportional to the intensity or the square of this projection. You projected something in that dipole moment of water a while ago. So that's where this came from. And it goes as the cosine squared. The intensity goes as the cosine squared. And for example, at 90 degrees, there will be none. All right. So the next section here transitions to the visible spectra. And as indicated in one of those previous figures, these involve low energy electronic transitions. And this corresponds to two basic kinds of biological molecules. It corresponds to metals, particularly transition metals, particularly what are called d orbitals, and large aromatic structures and conjugated double bond systems. Okay, so somebody asked me, how do you know it's copper? Okay, and so this is another way of thinking about it, that what's out there is going to be due to some aromatic conjugated double bond system, think heme, or it's going to be due to a metal. Okay, I don't know exactly what you're talking about with John at the moment, but certainly when you talk to John, what's his focus? His focus is entirely on the metal centers in all of the enzymes that he's interested in. And they all have very different characteristics, but they have very different electronic, vibrational, rotational characteristics, and they're the window into how those enzymes work. And so that's partially what this is all about. And then some systems are these aromatic conjugated double bond systems, think heme, for example, okay? So they say metalloproteins and conjugated structures like vitamins. Heme proteins owe much of their absorption to conjugated hemes rather than the metal iron, although the iron certainly contributes. All right. They're usually very broad bands because they consist of lots of subbands, lots of potential vibrational states. And so you don't get sharp bands like you do with oxygen or like you do with N2 or diatomic molecules, okay? So this broadening reflects the different electronic configurations. It also reflects different environments, meaning different binding sites or dynamics or conformational changes in the protein binding sites. Okay. So I've summarized what they've said in a slightly different way here. All right. So the visible region corresponds to transition metal ions. They're usually d orbitals, so they tend to have bright colors, think blues, large aromatic structures or conjugated double bonds. And they tend to reflect different environments and subbands. Now, the UV, which we usually describe as 2 to 400, would be smaller conjugated ring systems. And examples of these are tyrosine, tryptophan, phenylalanine, or purines and pyrimidines. So aromatic protein amino acids or nucleotide bases would correspond to this UV region of the spectra. These are quantum mechanical concepts, but these correspond to what are called pi-pi star transitions. So pi electrons are electrons that are involved in bonding. And in quantum mechanics, you also have what are called anti-bonding orbitals. They're configurations that basically don't interact, and they're known as pi star. And so you're having transitions. The light is being absorbed in these structures, giving rise to a pi-pi star transition. Go back up above to that figure where you have two different ground state, excited state, and the light shifting from the bonding to the non-bonding orbitals. Okay. So that's what you're doing basically in the visible. Now, the book has a table of the aromatics and the bases, adenine, cytosine, uridine, guanosine. Notice there's a pH dependence. So the spectra will depend upon the pH and the hydrogen bonding properties of these groups. It's showing you what the lambda max is. So you can see that tryptophan has a max at 280. Tyrosine has a max at 275. Okay. And then all of the bases have max that are around 260. And most of you know that you look at nucleic acid and you look at around 260 and you look at protein around 280. Maybe you don't know that, but you know that now. All right. And then it shows you what the extinction coefficients, how much light is absorbed. Okay. And so it gives you some property of how strong the I0 over I is, how big a change in the absorbance is per unit concentration. So notice the tryptophan signal is much bigger than the tyrosine, which is much bigger than the phenylalanine. So we will primarily use tryptophan and tyrosine for concentration. Notice all of the bases have comparable, not exactly identical, comparable extinction coefficients, but we'll have a little bit more to say about that in a moment. Okay. So I grabbed this because this is basically a restatement of what I've written here. Okay, so I sort of grabbed this, but then I restated it. All the purines and pyrimidines absorb strongly. Most of the absorption bands are pi-pi star. Okay. So I think my summary up above is what this is talking about. Notice the wavelength corresponds to the size of the conjugated system. Think particle in a box. Notice they're calling it the particle in a box rule. Okay. So it goes as 1 over a squared. Okay. And so the energy will go inverse with the size, and the wavelength, therefore, will be inverse with the size in that regard. Now, here's an example, and they refer to this example at the bottom of this section, corresponding to methylthymidine. Here's the extinction as a function of wave number. And it, again, is an oriented sample where it's perpendicular That doesn't look P and N. Let me look something up here because I'm not understanding. Oh, P and N has to do with certain, I'm sorry, P and N has to do with orientations on the figure. So it's parallel to PB this way, and it's parallel to P. So this is B and this is P, showing you that it's the P orientation where the alignment between the moment basically in the molecule corresponding to the electric vector in the polarized light. So this is where you get the maximum absorption. Think that cosine squared angle course, cosine squared theta corresponds to. And in this orientation, that's mostly zero. So there's very little absorption. Now in a solution, the molecules are tumbling around. And so it's sampling all of these various orientations. Okay, and so that's what this is showing. All right. So what I want to do now is a real quick introduction to what this means and how we use this. I usually don't give you guys a break. I just talk until you ask for a break. If I gave you a 5-minute break right now, do you want a 5-minute break? Yes, we're taking a 5-minute break, and then we'll come back to predicting extinction coefficients. I'm gonna get out of that for a second and open up something else to prepare for this. So 5 minutes. Okay. Are we back? Okay, so table 8.1 shows wavelengths and extinction coefficients. And then this is a summary of, and then here's a typical example of a spectrum. Okay, so this paper, Protein Science, 1995, so Nick Pace is the PI in this lab. He's at Texas A&M. He's still at Texas A&M. I'm pretty sure that Nick is still active. I don't think he's retired yet. How to measure and predict molar absorption coefficients of a protein. Okay, and so the absorption spectra of proteins is an important thing because we basically need to measure concentration, and we need concentration for all kinds of things, including, for example, quantifying enzyme activity. But just basically all the different properties that we need to know about a protein often begin with knowing how much you have. Okay, and so this is the abstract from that paper. I'm not going to go through the whole paper. I'm just going to basically do this. It's based upon the concentration measured by dry weight, nitrogen, or amino acid analysis. And so basically the way historically people measured concentration was to take a dry weight. Literally, you make up a sample. You add so much solid protein to a solution. Don't worry about, but you have to worry about how it's equilibrated in the buffer, so it's not just solid into a solution. And then you literally evaporate the sample and dry it down, and you measure the quantity left behind. So you have to measure the jar that you're going to make the measurement in without the solution, then fill it, and then dry it and measure the difference. It's a traditional, most accurate way of doing it. It is so accurate that you can't touch the vial. If you touch the vial with your fingers, you measure fingerprints. So you literally pick the vial up with tweezers so you don't actually leave anything behind. Okay, and you do this in multiple vials to get it accurate. It's a pain in the butt to do it. The amino acid analysis is another way of doing it. But what evolved over the course of many decades was a simple spectroscopic method that originally was developed by somebody called Edelhoff. It was not a quite linear path. And then an alternative method or a related method came out of Gill and von Hippel in 1989 that expanded this method. But this paper was sort of the next step and the final step in the technique. Okay, and it turns out the absorption of a protein at 280 depends upon tryptophan, tyrosine, and disulfide bonds. Okay, so if you have disulfide bonds, that contributes the tyrosines and the tryptophans. And what this paper does is it looks at hundreds of different proteins, 116 in the big table, and then asks how well you do by the method that they've arrived at in fitting the data and analyzing the accuracy of using this very simple method. And the simple method is shown in this equation. The extinction coefficient at 280 for any given protein depends upon the number of tryptophans, literally how many tryptophans in the sequence, times 5,500, plus the number of tyrosines times 1490, plus the number of disulfides, which is a cysteine, times 125. So you can see that the disulfides don't contribute much unless you have a lot of disulfides, all right? And so literally, if you just simply add up trip, tyrosine, and cysteines, disulfides, you get a reasonably accurate measure of the extinction coefficient, which then you can use for determining protein concentrations. Notice the mention up here of guanidine, hydrochloride, or urea. When you denature a protein, and they compared guanidine and urea and other solvents like propanol, part of the question is, what's the environment? And does that affect the spectra? And therefore, does the solvent that the protein is in matter? Does it matter that it's a folded protein versus a denatured protein in urea or guanidine? Okay, and so all of these are sort of considerations, and a lot of people in the field contribute to this, all right? But the general basic theory that comes out of this paper is that for folded proteins in water, this formula is a reasonably accurate way of predicting an extinction coefficient. Okay, so let me bury that for the moment. So there's a software package out there called Sedinterp, Sedinterp. And I've got an old version and a new version. I'm showing you the old version. So when it opens up, it has a database in it, and it allows me to choose a large number of samples that I have put into it. It doesn't actually have as many samples as I normally would have. And what I have here is human serum albumin with disulfides, okay? And so when I say compute, what it does is it reads in the amino acid composition. Notice that there are 17.5 disulfides in this molecule. And then down below, there are 28 tyrosines and 18 tryptophanes. All right. So that is how this software package calculates the extinction coefficient. When you read these sequences in, you can read them in 1-letter or 3-letter codes. There are 585 amino acids in this protein. If you add up all of those amino acids, correcting for the peptide bond, the molecular weight of human serum albumin is 66,437. Okay, so that's a number that we use for the molecular weight of serum albumin, human serum albumin. It neglects any post-translational modifications that may occur. Notice there's a V-bar estimate. We talked about V-bar in this class. Okay, the V-bar estimate, which is basically the average V-bar over all of the amino acid compositions, comes out at 0.7355. There's a temperature dependence to this that we'll talk about later. There's also a hydration that you get out of this, gram per gram, how much water per gram of amino acid or gram of protein do you have? So the hydration is something that will contribute to the hydration or the hydrodynamics of this protein. All right. Once you, and then if you had modifications on the protein, you could then add those and include them in the analysis. So for example, if you had sugar, lactose, or some other kind of sugar, or you knew you had guanidine involved, or there was glycerol binding or something else happening, you could include those as a cofactor. Okay. This software will also show you what the calculated charges are. So it's looking at the titratable groups, and it's telling you where the calculated isoelectric point is, 5.9. It's telling you what the total charge on the molecule is at pH 7, minus almost 13. That's calculated ignoring anion binding or salt binding. Okay. You can plot the charge and you can see that it's giving you a number at pH 7, but obviously it changes as you titrate all of the groups. Okay. It shows you something about the terminal groups and the calculated values for the terminal groups, because you know a little bit about that they have a different environment. And so they tend to have different pKs, making them more accessible, particularly the alpha amino. All right. But notice what this table also gives. It gives the absorbance properties in guanidine and the estimated absorbance properties of the folded protein in water. And it's using the method of PACE. The one up above uses this initial Edelhock method for guanidine. This is using the method for PACE. So what it says is that the molar extinction coefficient is 34,500. This is 3.45 times 10 to the fourth. Per mg per mil, it's 0.519. Okay. And so this software package basically uses the method of PACE. I use this technique to get a calculated extinction coefficient. In my experiments, I have different path cells. I don't just have 1 centimeter cell. I tend to actually have 1.2 centimeters or 0.3 centimeters. And so I adjust this extinction to correspond to the path length. Okay, so it ends up being extinction coefficient times L. All right, and that's what I'm using to convert the signal into a concentration. All right, and so that's basically what this software package is doing. This software package was developed sometime in the early 90s, I think. There's probably a reference if you go to the software and you look up the reference material to those initial calculations. There have been multiple versions of it. This is the newest version that actually I'm having trouble with, so I don't use it as much as the others. It's a little bit incompatible with the Windows that I have in this computer. It's also a little incompatible with the Windows I have in my Mac. But nonetheless, this is a software package that we would use for this purpose. Okay. Okay, so I stopped here because this table is telling you these are the characteristics, the spectroscopic characteristics of these amino acids. In principle, therefore, you can just sum up the sequences, the sequence aromatics using these numbers. Notice these numbers, it was 5,500, it was 1450, and it was, phenylalanine wasn't even listed because phenylalanine has a peak down around 250. So it has very little contribution to 280. And then the disulfides don't show up here, but those end up contributing a lot. Since there are 17 disulfides in serum albumin, it's 17 times 125. So they make a big contribution to the final signal. And I include it in all of my calculations when I work on human serum albumin. Okay, well, that's actually a system that I'm doing experiments on. All right. The other paper has to do with light scattering corrections. And so this is a Journal of American Chemical Society, JACS. It comes out of Harold Scheraga's lab. Scheraga's in his 90s. He might be 100 now if he's still alive. I saw him at a meeting 3 years ago or 4 years ago. He may still be alive. I don't think he's retired if he's alive. And it's basically correcting UV spectra for light scattering. And the sample that they're using happens to be insulin. It's zinc insulin, which has something to do with the association properties of the insulin. In the presence of zinc, insulin makes hexamers. So that has some contribution to the light scattering. This is a comparison of an absorption spectra for a particular sample in a 0.5 centimeter cell. It shows data from 250 to 350. You can see the peaks in the sample. So the resolution here is pretty good. It basically is showing you resolution of peaks that partially correspond to tyrosine, tryptophan, phenylalanine. But what it's showing you is what the raw spectra looks like in A and what the light scattering contribution is in B. So they've subtracted out all the light scattering. So when you take a 280 and you get a spectra at 280, it doesn't necessarily correspond to the peak, and the environment might change the position of the peak. So that's something that you have to worry about. But there should basically be no absorbance in a protein sample above 310, 320. And in this particular set of samples, they have a fair amount of optical density difference. They're actually comparing this by a difference method. And you can see that there's a lot of absorption out here, but there shouldn't be, because there's no chromophore in the protein that should be absorbing at 350. All right. So the method basically is a light scattering method. What you do is you take the logarithm of the optical density minus the logarithm of the optical density. This is something to do with the definition of absorbance. And you plot it versus the logarithm of the wavelength. Okay. And what you get is a curve that has a slope to it. You get the information in the region where you know there are chromophores, tyrosine, tryptophan. In insulin, there are, in fact, multiple disulfides, so they're contributing here. But then out here in this higher wavelength region, you're getting a change in the wavelength, the absorbance as a function of wavelength. This is all due to scattering, okay? This is all due to scattering. The wavelength scattering is proportional, generally speaking, to 1 over the 4th power of the wavelength. Okay, 1 over the 4th power of the wavelength is generally the way that we talk about it. The why is the sky blue usually uses the 1 over the 4th power of the wavelength in the narrative. Okay, and that's why you mostly see blue light, because blue light has smaller wavelength and therefore a smaller number, which therefore contributes to a larger amount of scattering. All right, and so the technique is simple. You take minus the log of the OD versus the log of the wavelength, and then you fit this part of the curve to a straight line. So you have to choose the data points that are on the straight line, and then you extrapolate those points indicated by the ... You extrapolate those points into the absorbance region, and you subtract them. Now, you don't subtract them in this mode. You have to subtract them back over here in absorbance mode. Okay, so you make this conversion, you get the optical information here, the wavelength dependence. When you do this, notice the slope is 2.5 and the slope is 1.9. So they don't get the 4th power, they get something else. There are all kinds of reasons why that might be. It has something to do with the shapes of the aggregates. The lambda to the 4th power minus 4th power is assuming spheres, small spheres. If they're not small spheres, if they're much bigger aggregates than the hexamers that I described, that would explain this. Okay, so the magnitude of the slope is potentially information about size and shape. A lot of the light scattering that comes from the sky is actually called me scattering. It has to do with scattering from really large particles, what's happening up in the higher atmosphere. That generally goes as 1 over lambda to the 2nd power. Okay, so it's not clear why this is what it is, but it's not important because it's basically a calculation to correct the data. It's not imposing theory, it's just being very practical. This particular sample has this slope, subtract that from the raw data. And when you do that, the scattering in the 3, 20, 30, 40, 50 goes away. Now the extinction, the absorbance that you get at these values is a true reflection of concentration. In A, this was concentration and scattering. You had no idea what the concentration actually was until you corrected for the scattering, okay? I would say that I'm the only person in this university who corrects for light scattering, because most people aren't that concerned with their concentration. One of my students a few years ago, Valeria, who some of you may have known, probably don't, because she was graduated quite a while ago, was doing experiments, and it was quite clear that there's something wrong with her data. The data didn't go through the 0 point correctly. It should have gone through a certain point at 0, and it didn't. And it suggested that there was something wrong with the concentrations in her plot of density versus concentration. It should have gone through the point of the density of the buffer, and it didn't. The extrapolation was wrong. So I said to her, correct for light scattering. And she said, how do I do that? And I said, Scheraga, 1960. She read the paper, she corrected the data. Now the data went through the buffer density on her ordinate, and she was getting the correct value now, okay? So it's something that happens in certain samples, and it's something you have to correct for. So I've given you a spectra in the assignment of one of her samples that has light scattering in it, and I'm asking you to correct for it. This is the method. I've outlined it for you. The paper is only about 3 or 4 pages long. It has some other details in it that may or may not help. I think what I showed you is the gist of it. All right. Now, I've introduced you to extinction coefficient calculations for proteins. I've introduced you to scattering corrections. Can we do the same things with nucleic acids? And the answer tends to be no. Sometimes, okay? And the problem is that absorption in nucleic acids is much more dependent upon the structure. Is it monomers, dimers, oligomers? So is it a strand of single-stranded? Is it double-stranded? What is the structure of the nucleic acid? And it's shown in this particular figure from Van Holde, molar extinction coefficient versus wavelength. And what you can see is that AMP alone has a peak around 260. Okay, that table previously says it's around 257 or something. All right. Single-stranded poly A has a slightly shifted peak, but also a smaller extinction. Double-stranded DNA has a slightly more shifted peak and less absorption. So these are reflections of what are known as hypochromism. It's a change in the absorbance depending upon the nature of the polymer and of its molecular state. Is it single-stranded poly A? Is it double-stranded poly A? Okay. So it differs from that of a monomer, and the intensity and the peak position both change. And so how do you do this with this kind of calculation technique that I just described? It becomes much more difficult to do it by the technique. This is another reflection of this. This is a melting experiment. This is double-stranded DNA. It's showing the absorbance as a function of temperature compared to 30 degrees. And as you heat the sample, there's no change, there's no change. And suddenly there's this huge increase in the absorbance. As you get above 80 degrees, the midpoint is around 85, 86 degrees. And so what we're doing here is we're melting the DNA, we're breaking all of the hydrogen bonding. And as we do so, the absorbance goes up. All right. The absorbance goes up, and therefore single-stranded DNA absorbs much more than double-stranded DNA. Okay, on this previous figure, it looks like a small change here for the single double, but this has to do with a mixture of something. It doesn't tell you what the DNA is. So they point out a couple of things. The first thing is that it's sharp. So that typically means that it's cooperative. Okay, once the double strands begin to break, they all break. All right, and it refers back to chapter 3, which has to do, by the way, with the binding interactions that we talk about. And then the other thing to notice is that as you cool it, it doesn't follow this same line. It follows a completely different line back to the baseline. That's because, as you probably know, when DNA reanneals from its single to its double-stranded form, it has to hunt, it has to search, it has to find the right alignment to get everybody in the right position to completely form the right double-stranded sequence. Okay, that usually takes time. That usually takes long incubation. Generally, you wouldn't stop at 30, you'd go all the way back to ice, and you would let it sit there a long time to make it completely reform the double-stranded form, okay? But again, can you use absorbance to measure concentration with single-stranded, double-stranded DNA? This kind of a data suggests that it's sort of difficult, okay? It's sort of difficult. Okay, so we're into a slightly different explanation for that, and they're trying to explain this sort of in terms of, again, of transitions. But the fact is that when you put two chromophores near one another, so imagine these are two bases in DNA, and when they're near to each other, they might be oriented in a slightly different way, and they're interacting with one another through dipole-dipole interactions. Okay, and therefore it causes a splitting in the banding pattern in the energy levels, and you can have transitions between ground state and activated state in multiple forms. This changes the wavelength, this changes the intensity, and so this is part of an explanation for why the absorption depends so much on the state of the bases: monomers, dimers, polymers, single-stranded, double-stranded. Okay. And so generally speaking, when you do a concentration, you want to make sure in nucleic acids that you collect the data at a certain temperature so that the sample is completely denatured and you're making your measurements under denaturing conditions. But generally speaking, people don't trust it that much for absolute concentrations. It becomes difficult to do. Okay. Let's briefly talk about fluorescence. So in the previous figures, when there was a transition from ground state to excited state, the energy going from A to B was what you observed. In some molecules, B is got a lifetime, and it decays to a different level. And it decays to a different level, C, over some time, in such a way that the light that is emitted during the excitation of that particular sample is at a different energy and therefore a different wavelength. Okay, so the absorbance corresponds to A to B, but then this state is not stable. It relaxes to C, and then it re-emits the light at a different wavelength because it's a different energy going back to D. So in fluorescence, the absorption spectra looks like this, but the fluorescence spectra is shifted to higher wavelengths, lower energy, for the reasons that I just explained here in the figure. This fluorescence spectra is usually very characteristic of this lower energy state, and it almost doesn't matter where you excite. You can excite down here at 250, this is the spectra you'll get. You can excite at 270, this is the fluorescence spectral you get. Because regardless of where you go up here, it will relax to C, and that will always be the transition you get when you re-emit the light, all right? And so the fluorescence signal is usually a very characteristic measure of what's going on. In addition to that, different chromophores within a molecule can interact with one another. And when they interact with one another through dipole-dipole interactions, you can get a change in the re-emission from that chromophore that reflect the fact that there are interactions. Okay, and so this interaction can also be reflected as what is called an overlap of fluorescence spectra of donors and acceptors. So for example, in the text, I think it mentions, in the text, it mentions tyrosine tryptophan. You can activate through a wavelength that activates tyrosine, but what you'll get is fluorescence from tryptophan, because the energy always ends up going to a tryptophan that's close by. And so it can literally take the energy and give it up to the tryptophan. The efficiency of that goes as 1 over the distance to the 6th power, okay? Another way of saying that is how efficiently this occurs tells you how far apart tryptophan and tyrosine are in the molecule. So the dipole-dipole interaction depends upon the distance between the groups, and that means that this inverse 6th power separation is a ruler. It allows you to measure the distance between the tryptophan and the tyrosine, or between whatever the donor and the acceptor molecule are. All right, and so in this particular example, if you are exciting tyrosine, you would always get the fluorescence spectra of tryptophan if they were close enough to one another, and close enough to one another is 20 angstroms. Okay. So just one figure showing you the general design of a fluorimeter. We have a fluorimeter in my lab. You have polarized light going into a sample. Okay, the orientation of the light matters. And there are ways in the design of the instrument to accept light in only one direction and analyze light in other directions. But then you look at the re-emitted light at right angles. So the cell actually has a face that allows light to go through it, but then a side face that allows the fluorescent light to come out. And that's where the detector is. And so the emitted light, the absorbed light comes in this direction. And then there's a right angle basically that allows you to measure the fluorescent light. And that's the information you can get. And you can measure parallel perpendicular. What that gives you is some information potentially about how quickly molecules reorient. We're not spending a lot of time on this because this chapter doesn't talk about it a lot. This is as much as I'm going to say about it. It's described in far more detail in the newer Van Holde. Okay. So the next topic, and we only have about 15 minutes, so we're not going to get very far in this probably. Not as far as I thought. The verboseness of your lecturer. So one of the things about biopolymers, about macromolecules, is that they often have helical secondary structure. For example, alpha helices in proteins, single, double, triple helices in polynucleotides. There are helical conformations in polysaccharides, etc. But absorbance and fluorescence don't necessarily see that. Okay, I just described to you how it affects absorbance, but it's not necessarily easy to relate it to the nature of the interactions, because it's not really, it's just showing you absorbance is up or down, but and slightly shifted, but it's not generally easy to do. Okay, and he makes a short argument here as why it's not necessarily the ideal way of doing it. And the argument is that we need a physical property that explicitly is sensitive to asymmetry of the structure, the helical nature of the structure. Number one, is it helical? Number two, is it left-handed or right-handed helical? Okay. And so there's a fairly long introduction to the properties of light in this chapter. So electromagnetic waves have an amplitude, and if they're polarized, that amplitude is in one direction. So in this case, it's in the Z direction. Okay, so the light is coming along the x-axis. The source is back here. The light is coming along this direction, and the amplitudes go in the Z direction. And it's a wave, so it's high, low, and there's a certain wavelength in terms of separation between the peaks. The electric vector induces a magnetic vector that is perpendicular to the electric vector. So the electric vector is usually called E, and the magnetic vector is usually called H. Here we go up here. So this is E, and this is H. And it's always perpendicular. Okay, so there's an electric magnetic component to light. The electric vector induces a magnetic vector. Okay, and this describes, I think, what I've just said about the nature of this. The oscillations occur with a frequency V, which is inversely proportional to the wavelength. However, light doesn't have to be polarized in a single direction. Light can actually be circularly polarized, so that the light is actually rotating as it goes along the path from the source to the direction that it's going. So this is right circularly polarized light, and I think you can see that it's rotating from out of the plane, into the plane, out of the plane of the board or of the screen. And so this is circularly polarized light, and this is particularly right circularly polarized light. And so you can make instrumentally circularly polarized light. So now that you have circularly polarized light, what might one do with that? All right. So this is showing this in more detail, that you can have right circularly polarized light or left circularly polarized light if you combine them in equal quantities, equal amplitudes. The amplitude corresponds to the size of the E vector. Then these two waves would cancel one another and produce light polarized in the average dimension. Okay. However, if the left and the right circularly polarized light were unequal amplitudes, then that would create a resultant light beam that was right circularly polarized. It would be the sum of the projections of these two vectors onto the resultant. And so you would get elliptically or circularly polarized light out of the combination of these two left and right circularly polarized beams. All right. That gives you a hint as how you might make light like this. Okay. And so that's the first thing. You can make circularly polarized light. So if you have two plane polarized waves, but they're not quite in sync. And what that means is that the peak of E1 corresponds to the minimum, not the minimum, corresponds to the 0 point in E2. So they're out of phase. And when you create two beams, polarized but out of phase, the sum of these vectors will give you circularly polarized light. And so the text, or my version of the text, is explaining it here. Represent circularly polarized light as the sum of two plane polarized waves, 90 degrees out of phase, with their electric vectors perpendicular to one another. So this one is in the z-axis, this one is in the y-axis on that previous representation. All right. So white light, unpolarized light, will have no fixed direction. It will be what we call incoherent. In the instruments that we're talking about, we make polarized light by putting in some kind of a device that selects for only light in one orientation. And you can rotate these polarizers and analyzers to select the light that only has that polarization. So that's how you do it. Okay. And then you create slightly out of aligned phases so that the light sums in a way to create circularly polarized light. All right, so that's an introduction sort of to the light. So now what? How does this polarized light interact with molecules? And there are two ways that the light interacts. And these reflect what is referred to as optical activity. They either express optical rotation and or they express circular dichroism. Okay, and so it's described in the legend. Okay, CD bands can be of either positive or negative sign. So what that means is that circularly polarized light can prefer left or right circularly polarized light. And so they will selectively absorb one or the other. And in the definition of delta epsilon, it's the extinction of the left minus the extinction of the right. The extinction of the left and right depend upon the direction of the helix. Is it a left-handed or a right-handed helix? And it will see the light in a slightly different way. And therefore, it will be a positive or a negative band that corresponds to the direction of the helix. Okay. The delta E is small, much smaller than the extinction. So it takes a lot of sensitive equipment to make this happen. This example is showing you methylcyclopentane. It's showing you an absorption spectra. It's showing you a circular dichroism spectra, or a delta E. So it's showing you that sometimes it gives you a positive signal, sometimes it gives you a negative signal, and that begins to be a window into what the structure of cyclopentane is. In addition to positive or negative, some bands will be intense and some will be weak. Okay, and he points out the 178 band here, where you have a quote unquote strong band here, but a weak CD signal here. Okay, and so this particular technique is sensitive to the structure, but it's sensitive in a complicated way. It's not just that it might be positive or negative peaks, it might also be weak or intense. And so there's information in a CD spectra that gives you information about the symmetry of the structure in the macromolecule. It's not just the concentration of tyrosine tryptophan. It's not just the presence of a 260 peak. It's also giving you explicit information about the tertiary structure the secondary and tertiary structure of what's going on. Okay, mostly secondary in that example. In addition to circular dichroism, there's something called optical rotation. And this is described in this figure, where there's a wave of light coming in oriented in the Z direction and goes through a sample, and the light emerges at a different angle. It's rotated by interaction with the sample. The amount of rotation corresponds to the concentration, the sample thickness, that sounds familiar, and the specific rotation of the intrinsic molecule. The rotation can be either plus or minus. This corresponds to dextrorotatory or levorotatory in the chemistry notation. Okay, so the light will be rotated one direction or the other, depending upon the symmetry of the macromolecule or the symmetry of the molecule. And from the rotation, it tells you something about not just the presence of asymmetry, but also the particular direction of the asymmetry. All right, now, this is going into a little more definition of what this is. So I'm going to stop here. And I'm going to stop here. Okay. So where we're going to go with that is relating optical rotation and circular dichroism to basically particle wave characteristics, and basically refraction of light, the scattering of light versus the absorption of light. Okay, one of them gives you something about scattering, one of you gives them something about absorption. Both of you give you something about asymmetry. One of the easy examples of this is, and I think it's used as an example down below in the PowerPoint. You know that all the amino acids in proteins are L amino acids. They're not R amino acids. In bacteria, we use R amino acids for channel sometimes, synthesized in a completely different way. So all the asymmetric centers in amino acids, 19 out of the 20 common amino acids are asymmetric centers. The carbon is an asymmetric center. It has an L configuration. Think levorotatory, dextrorotatory. They will cause rotation of the light in a certain direction because the asymmetry of the carbon is in a certain direction. And they will have a minimum in their CD spectra or a maximum in their CD spectra because it reflects the orientation of this the asymmetry of the central carbon. If you make up buffers with L histidine, you cannot do CD on that sample because the CD spectral will be the buffer. You have to use a racemic mixture of L and R histidine so that they cancel out, and then you can see the protein. So you have to actually be careful about the buffer that you choose in the background of the protein, otherwise it messes up the measurement. Okay. All right. So we're not done. We're at 34. I think there are 43 slides here. Okay, so hopefully we'll be able to get through those 44 slides. We'll be able to get through those slides in the first hour next week, and then we'll do what I said we were going to do about reviews and going over certain kinds of examples. The assignment has some questions from chapter 8. It has some questions from chapter 9, chapter 10. I think it's 5 total questions there. And then the one question about light scattering correction for 6 total questions. We're not done, so the assignment doesn't happen until next week. Okay, so it won't be due next week. It'll be due the week after. Questions? Any questions at this point? Okay. I'll see you next week. Thank you.
Thank you, Dr. Correa.
Thanks, Dr. Correa.